Thank you, everybody, for coming. Okay, this is lecture number 2 in our molecular biology course for this year. And our speaker this morning is going to be Michael Patnode. So Michael grew up in the Seattle area and went to the University of Washington, of course.
That's the place to go.
But then saw the California light and got a PhD at UC San Francisco. But couldn't stay away from Washington, but got confused and ended up in the one that's in St. Louis. Washington, right, which is Washington University, not to be confused with the University of Washington. All right. And that's where he did his postdoc. And he came to Santa Cruz in 2020 and is finishing his 4th year here, and it's going really well. I give this course on food and biochemistry, and I've been talking about why you need fiber in your diet, and got me into reading about our microbiome, which has just been an explosive topic in biology in the last 10 years or so. And now we've hired somebody who really knows about this. Michael.
Well, thank you so much for that introduction, Barry, and thanks everyone for coming. Thanks for the invitation to share some of what we're really excited about. As Barry mentioned, I started my lab in 2020, and we work on diet and how it interacts with the bacteria that live in our intestines. That's our main area of focus. We want to know what makes so-called healthy foods healthy, what encourages good bacteria or beneficial bacteria to grow in our intestines versus bacteria that might cause problems. And we also want to know about the immune system and diet, and how does what we eat impact our immune system. So I'm going to be talking about a lot of that stuff today. We'll kind of go in 2 parts. So first I'll give like a overview of microbial communities and how bacteria mainly work together with animals that they colonize to improve the health of both of those members of that partnership. And then I'll spend if we have time at the end, I'll talk a little bit about some new, what we feel exciting data coming out of our lab that's unpublished and has to do with the foods we eat. Yeah, and then I would love it if people would interrupt me at any time with questions. I hope it can be very interactive, and I can make sure that I'm explaining everything properly. So yeah, definitely just shout out or raise your hand. I'll try and catch people as they have questions. And if somebody's monitoring the Zoom,
I am.
Okay, great. Yeah, if you have questions and you're zooming in, go ahead and type them in the chat, and I'll try to get to those too. So I like to start these type of discussions with this image. I think it's so fascinating. This is an image of dental plaque from a human donor. So it's a microscopy image. So it's shown much larger here than it is in real life. This is about 100 microns across, so barely visible to the naked eye. And this was taken from a healthy adult human in the US. The colors are individual species of bacteria. So I like the image because it illustrates how many different species live on our bodies, especially in places like our mouth and our intestine. The shapes also give you an idea of how diverse bacteria can be. There are sort of round bacteria in this image. Those are probably cocci, and then there are rod-shaped bacteria, which you might be familiar with, like E. coli. And then there are these long filamentous bacteria that are very distinct from the rod-shaped ones, and the cocci. And you can see that in some cases, certain species appear to associate with each other. So this teal color and the pink color are often found closely arranged. And then the orange and the green here are often together as well. So we find this idea that different species might be interacting with each other in a reproducible way, or that they might have some relationship that they've evolved. We find that to be really interesting. And although there's a lot known about the oral microbiota, which is present mostly in dental plaque and on the surfaces of our tongue. There's not as much known about the gut microbiota. And so that's why we've chosen to study that in the lab. Any questions about this image?
You know what all the species are?
Yes. So the answer, amazingly, is yes. And I'll talk a little bit about that in a few slides, how we could possibly take a complex community and understand every single type of species that's there. We can do it with DNA sequencing, which has really only been possible maybe starting in 2000 to 2005. The advent of next-gen sequencing has allowed us to really catalog all of the species that are present in the sample. We don't know what all the species do. It's sort of like initially stamp collecting. You sort of find all the different members of this community, but actually figuring out which ones are harmful, which ones maybe cause cavities, which ones maybe compete with microbes that would cause cavities. Is there a beneficial species that maybe would outcompete cavity-causing microbes in the mouth? Those relationships we don't understand just from sequencing. So that's where all the hard work comes in.
Is this under the biofilm that I've heard they make?
Yeah, I think you can think of this collection of species as making up the biofilm. So this would almost be an image where the cells, each individual cell, which is a bright color in the image, would be suspended in a biofilm, what we call matrix, which is invisible in this image. So bacteria in a biofilm is kind of like chocolate chips and chocolate chip ice cream. They're just, suspended in that medium. And so that is likely what you see here. Yeah.
Michael, I think for this group, we're gonna have to speak out just a little more.
Oh, gotcha. I will try.
I told them.
I shouldn't have said for the.
No, you shouldn't. That and she's done. This is from dental plaque. And that's what we're supposed to offer of our teeth, so I assume it's not good for the teeth.
Right. So in the mouth, the consensus currently, although we don't understand the precise contributions of each of the species in our mouth to the formation of cavities. What is clear from the studies that have been done is that the overall amount of bacteria is correlated with dental cavities. And so the advice of clinicians and dentists is to remove as much of the bacteria as possible, which gives rise to, intense brushing and things like Listerine that kill, bacteria in the mouth. Yeah, great question. Yeah.
How did you get all those beautiful colors?
Yeah.
They aren't colored like that, right?
No. Some of them do have interesting colors, so you can isolate the species from the mouth individually and grow them individually instead of in a community of microbes. And they do have interesting colors. These are not the natural colors. The colors are the product of staining each of the species with a unique, it's actually a nucleic acid barcode. It's a DNA, a small stretch of DNA that matches only one species type in the mixture. And then you can attach to that deep piece of DNA a colorful molecule, a fluorophore. And so that's how this image was done. This is not my image, by the way. I want to make sure to credit Jessica Mark Welch and Gary Borsi at the Woods Hole in Massachusetts for these studies.
Okay, so back to the biofilms, that's mostly water, right? But what else? There's something else mixed in there.
Yeah. So, right. So biofilm does have a lot of water in it. It's, meant to, that's one of the benefits of biofilm. It keeps the organisms moist in that environment. There's a lot of polysaccharides, which is a long sugar molecule that the bacteria produce themselves. Kind of like slime, like a slug slime, for example, is contains polysaccharides like that. And then there are also proteins with different functions in the biofilm. And then there's free DNA as well, because it turns out that long stretches of DNA that have been released from dying cells are also very sticky and viscous. So that DNA helps to create this sticky glue.
And then we have the saliva,
True.
so we have an environment that these bacteria are invaded in.
Exactly.
It has its own immune properties.
Yeah, thank you. Thanks for bringing that up. So yeah, this biofilm exists stuck to the teeth normally, but if this region is stuck to the tooth, this region would be exposed to the saliva and everything you eat. So the bacteria take advantage of the nutrients in the food you eat, mostly sugars, simple sugars, which is why sugar is so detrimental for your teeth. The bacteria can suck up that sugar from the environment in your mouth. But you have in your saliva many immune factors that kill bacteria. So your body produces molecules to try and control the amount of bacteria. Yeah, thanks for bringing that up, because a lot of these questions that we're discussing are relevant also in the intestine. So the bacteria in your mouth, it takes, a day or two to start to notice that it's actually there. You can feel it on your teeth. That's bacteria growing on your teeth. But it's a small amount relative to your total body size. The bacteria in your intestine is usually 3 to 4 pounds of bacteria living in your intestine. So that is vastly more microorganisms than in your mouth, of course. So the complexity, the numbers of species, and the interactions with the immune system are sort of amplified in the intestine, which is why we like to study that organ. Okay, so for the first part of the lecture, I like to break this down into 4 basic questions about how animals and bacteria might interact with each other. So the first question deals with the benefits. Why would organisms evolve to have interactions with each other? Why would bacteria evolve to colonize our body surfaces and body cavities? I like to then kind of talk about the habitats, the ecosystems on our body that bacteria can occupy. The species, the actual individual types of bacteria on our body constitute communities. And you can think of that as understanding who is there on our skin, which species are there, armpits, mouth, intestine. There's different communities of microbes in each location. And then the last thing we'll talk about is the dynamics of how we interact with bacteria. When does this happen? It turns out that we are born with no bacteria. There's no other microorganisms in or on us in utero, in the womb. And so it's only when we were born that we get colonized with these species. So that's a massive change to our physiology when that occurs. And there are many groups interested in the dynamics of that process. When a baby's first born, which species come in, which species then replace them, and how do those species change throughout the life of that person? So we'll touch on that a little bit. Okay, so starting with benefits, we can think about this in terms of evolutionary advantages. And ecologically, we can actually quantify this concept. So if you think about these 2 axes, we can plot individual bacteria, mammal in this case, let's use a mouse as a model, individual outcomes of a bacterium and a mammal associating with each other. So if you take the fitness of the host without the bacteria, and then you add in the bacteria, and if the host is now more fit, if the health of the animal is improved, you can plot it on a positive axis, which we can call I here. And then you can also ask, when you mix that host with a bacterium, does a bacteria do better? is it beneficial for the bacteria to colonize the host? And if so, you can plot that as a positive on the J-axis here. So let's just take an example here. Let's say we're going to colonize a mammal, mouse or human, with species 1, which I drew in red here. Species 1, when it colonizes a mouse, the mouse does better, the bacterium does better. So this is called, which you may have heard of, symbiosis, or mutualism is another way to think about this concept. So mutualism is beneficial for both parties in this relationship, right? And there's a lot of examples of this. And in biology, these are really fun examples to think of because competition between organisms where they're trying to kill each other is so widespread and obvious in many cases. Mutualism is really special. It's an arrangement where the two organisms both benefit. Okay, so that's certainly the case for some species in our gut microbiota. And we have some examples of those that we've learned about over the past 20 years of research on this. There's a second type of relationship, which we can also plot on these axes. So species 2, totally different bacterial species in the intestine. Let's consider that it benefits a lot from colonizing us. It wouldn't survive in the environment, but when it colonizes our intestine, it can do very well for itself. That species, based on these axes, provides no benefit to the animal that it's colonizing. So that situation is called commensalism. It's a harmless bacterium. And there are actually many bacteria in our own intestines that we assume, or our evidence suggests, that they're commensals. They live in our intestines because they can do well there, but they don't provide any benefit that we can detect. Okay, and then the last sort of relationship that I wanted to cover is over here. So you may be kind of putting it together now. This bacterium does really well by colonizing the mammal, but the mammal does much worse as a result of being colonized. So this is, ecologically, is called a parasite. This is something that detracts from our health, but benefits itself by colonizing us. And you can also name this type of relationship pathogenesis, so pathogens, many pathogens, which are bacterial infections, could be thought of as parasites. They benefit by inhabiting our bodies, invading our bodies, and we suffer as a result. So I like to point this out because it really demonstrates why we might want to know what each of the species does in a place, a habitat like our intestine. Oh, sorry. Any questions on that sort of concept?
Are there any species that are negative on the J-axis?
Yeah, that's a great point. Yeah, so you could imagine maybe a.
Repeat the question?
Oh yeah, I will repeat the question for the Zoom audience and for the audience here. So the question was, what about the bottom half of this mathematical plot here? Are there species that when they associate with a mouse, they do very poorly? That's an interesting ecological space. We have less examples of where that would be true. I think you can think of maybe an accidental association. So if you consume food like lettuce or tomatoes from the garden, and there are a bunch of microbes on that food, they may enter your body. Your immune system, if they're not adapted to you to colonize your gut, your immune system may rapidly destroy them. It may just eliminate them from the body. So their association, results in a negative for them. The fact that they've made it accidentally inside your body is bad for that collection of organisms. Now that may cause inflammation for you, in which case it's bad for the host as well. I think more likely it's probably here where it doesn't affect you at all in that case. But yeah, thanks for pointing that out. There's a lot of interesting ecology here as well.
Question.
Yes.
Back to the commensal and mutualistic organisms. Could the commensal be beneficial to the mutual bacteria?
Yes.
And therefore they survive well and then the host survives well.
Yeah, thanks for the question. So because this is 3 species, in your intestine you have hundreds of different species of bacteria. And because those bacteria interact with each other, they're in the same location. They, in some cases, physically touch each other. And if not, they secrete products that can interact with other cells in the environment. Those bacteria can alter each other. So precisely, so you can kind of ask questions like, this commensal bacterium, maybe it doesn't directly benefit us when we study it in isolation, but if it benefits a mutualist, perhaps if we're designing a probiotic that will, somehow enhance our health, we would want to include the mutualist along with the species that support that mutualist. So yeah, that's a really important concept. And many companies who are designing they're called next generation probiotics, they're considering what species should be included to promote the individual species that maybe produce a beneficial molecule, for example. Yeah. Okay. So this is just to demonstrate some of the complexity. We won't go through all this, but the question about what are the other relationships, those can be mapped down on the bottom half of this type of plot. So this is sort of a mathematical space that ecologists use to study any 2 organisms and how they interact. So you can see predation is also technically in the same mathematical space and when a wolf and a rabbit interact, it benefits the wolf and not the rabbit. Okay. Okay, so those are it gives you an idea of how a microbe might benefit from associating with us, either to our detriment or maybe in a way that benefits us. But where do these benefits to the bacteria actually manifest? What are the habitats on our body that provide benefits to bacteria? So there's a couple of examples. So this, I like to break this up into outside surfaces and body cavities, because there are some distinct benefits of body cavities that make them a very attractive place for bacteria to colonize. So this is a humpback whale, and you can see that on the whale are barnacles, right? So these are not microorganisms, but they do colonize a host. So the whale is the host to these barnacles, and the barnacles get benefits from colonizing the whale. They get to be distributed, they get to be dispersed throughout the environment. They get to be carried to regions of high nutrient density. So the whale is like seeking out food and the barnacles are brought to the food. But they're also exposed, which is why they need this hard shell that they produce. They're exposed to a lot of stressors in the environment by colonizing the outer surface of the whale. So this, you can think of this on the human body as well. So the surfaces of the body are the skin, right? So we do have microbes on our skin, but the number of microbes on our skin is vastly less than the microbes that actually exist in our body cavities. And we're going to talk mostly about the intestine today. So you can think about a body cavity like the mouth and intestine as just a long hollow tube that is, it does have an opening at both ends, but it is protected from the environment once you're inside this tube. Okay, so I thought maybe it would be a good idea to just brainstorm a little bit, because every time I do this, I learn something new from the audience that I'm presenting to. So even if you haven't really thought about this idea before, what could you imagine might be some of the benefits of living in a body cavity, like our mouth or our intestine, versus living on the surface of our body?
Save some showers.
Yeah, exactly, right. Right, especially for humans. Yeah,
Temperature,
yeah, temperature stability.
pH.
Nutrients. Passing through.
Yeah, the food is provided is a big one. Yeah,
Moist-
temperature.
Moisture.
Yeah, moisture. Even though something like a shower or something like washing may be detrimental for the outside of the body, moisture on the inside, moisture in a protected location could be really beneficial to the bacteria.
Friends.
Oh, yeah, that's a great one. So if two microbes need to cooperate in order to thrive, that's a great place for them to always find each other. They're kind of in a contained environment. Yeah, any other ones?
Protection from light. From protections from ultraviolet light.
Oh, yeah, that's an interesting, I don't think I've heard that one before. UV light. Yeah, in organisms that are transparent, like tiny fish or little crustaceans, maybe that's not a benefit. But as soon as you get into a big enough size where the inside is protected from radiation, that could be a benefit too. Was there one more?
I was thinking of, like a sewage system. Disposal of wastes.
Oh, yeah.
Now, granted, a lot of those bacteria in our own disposal system just go right on out, but there must be some system in there that regulates, the buildup of toxins.
Yeah, I like that idea too. I had not thought of that one either, but I think that could be true. So, for example, when you grow bacteria in the lab, usually you grow it in just a tube of liquid, a test tube, and eventually the bacteria stop growing. One reason is they run out of nutrients. The second reason is they've built up waste products that become toxic to the bacteria. But in the intestine, since we have sort of a linear flow of nutrients in and waste products out, if bacteria are able to affix themselves to the intestine, they may benefit by the waste products being pushed out. Yeah, that's a great idea. Yeah, thanks for those ideas. I think many of these are considered to be established already. So certainly temperature, the warm temperature of a mammalian body cavity is a benefit. The neutral pH, the constant pH, the moisture that's present, the fact that there's other bacteria there, there's examples of these in the gut microbiota. I'll have to check out some of these other ideas, which I think are really creative and interesting. Yeah, and I mean, that would be really great to do experiments to try and figure out whether UV light is actually something that microbes in the gut are protected from. Okay, so all of these benefits, I think most or all of them, I would say are really very potent when the bacterium is inside a body cavity. And so that turns out to be where we have the maximum number of bacteria. The skin, so this has been determined through human studies, sampling different body sites and checking how many bacteria are there. And the skin is one of the locations where the biomass is actually lowest. And then airways, the mouth, and the urogenital tract, especially the vagina, these body cavities harbor many more species. And then the maximum number of species is in the intestine. That's where we have the most bacteria. Okay, so if these advantages are so potent and they've caused us to be colonized by hundreds of species of bacteria, trillions of cells, if you can imagine, why wouldn't bacteria actually just go ahead and invade our tissues where the benefits might be even greater? Sometimes they do, right?
Defenses.
Right, yeah, immune defenses. Is that what you're gonna say too? Right, so as soon as bacteria enter our tissues, they kind of leave this cavity area and actually get into our blood, our muscle tissue. They do benefit a lot, but they start to detract from our own fitness. They would move into that sort of left side of that plot that I was showing before. They become pathogens, and we have a strong incentive to eliminate them at that point. So, and we do, if our immune system is working well. So we can destroy pathogens that get into the tissue, and we, in general, tolerate most bacteria that reside in the body cavity. So it's sort of a sweet spot or a Goldilocks zone for the bacteria.
But we've got that barrier too.
True,
It's a wall.
that's true too.
And it's and that's, yeah.
Yeah, that's another really important, some people would even say that's sort of like the first immune system, is that you have a wall that makes it difficult for bacteria to get across. As I'll talk about, I think in the next slide, we can't make the wall too thick because we need to get nutrients from our diet. So especially in the intestine where, you know, our fitness depends on our absorption of nutrients from our foods, that wall ends up being pretty thin in most cases. And so bacteria, if they have a strong enough incentive, they can break through that wall and invade our tissues.
I have a question.
Yes.
Are there any, in the intestine, selective, or, you know, targeted, antibodies or defenses against certain organisms, particular organisms within the intestine?
Yes,
That our body.
absolutely, yeah. So in the what's called the lumen, or the interior of the of this tube, the intestine, we secrete our own cells, our human cells secrete molecules into that space that kill bacteria. It happens mostly in the early parts or the upstream parts of the intestine. And what we secrete are often called antimicrobial peptides that kill bacteria. We also secrete antibodies that are similar to the antibodies you make when you get vaccinated, and they stick to bacteria and interfere with their ability to grow. So yes, those molecules are made upstream usually, and they can control in some ways which species are allowed to or are permitted to thrive there. In the lower part of the gut, many of those antimicrobial peptides are not as concentrated. They can be broken down. And so that's actually what I'll talk about in the next slide, is that most of the bacteria are present in our colon and rectum. They're in the lower part of our intestine. Yeah.
Doesn't where you live and what your body's acclimated to also have something to do with which are good bacteria or which our body can handle?
Yeah, I think that idea is really exciting to people right now, because if we can understand how different people, individuals or groups of people are different in terms of which bacteria they have and whether those make them more healthy or not, we can more effectively practice personalized medicine, or medicine that's very tailored to a particular person's microbial community. Yeah.
Do white blood cells also invade the lumen?
Yeah, that's a interesting and somewhat controversial idea. So
What
there
is
are.
the idea? What?
Sorry. The question was, do white blood cells, immune cells that kill bacteria and circulate in our bloodstream, do they ever enter this luminal space? Do they leave our body and enter this body cavity? And what would happen if they did that? Usually the answer is no. Usually those blood cells don't survive in our intestine or in our mouth or in our other body cavities. But there are some studies, more recent studies, that suggest that in some cases, certain white blood cells can actually enter the lumen, but it's a little bit controversial. Yeah.
So, we have tonsils right up here. And food, water goes right by them. Do they play a role in this system?
Yeah, that's a great question. So there are a number of immune cell tissues or organs. Tonsils is one. And there are also immune cell organs that line the gut that are called Peyer's patches. All those immune cell, it's like collections or cities containing many different types of immune cells all together. And the idea is that in those locations, pieces of bacteria, pieces of food that you eat, small molecules from those sources actually get taken up by your immune system. And what happens after that is very complex, and it can result in what's known as tolerance, where your immune system learns to tolerate whatever's passing by. Or in some cases, especially if there's inflammation, it results in an immune response, where your immune system attacks or learns to attack whatever is passing by. Yeah. Okay, so these are some of the habitats, and I hope I've convinced you that the habitats that provide all these benefits are body cavities, and the gut in particular provides a lot of these benefits. I think the most impactful benefit that bacteria get from colonizing our intestine is the provision of nutrients. So I wanted to spend a little bit of time talking about the nutrients that we consume and how they benefit bacteria. So you are maybe aware that a lot of the nutrients that we get from our food come from the fat, simple sugar, starches, and proteins in our food. This is what's on the nutrition label. This is where we derive benefit from those foods. And those components are broken down and absorbed in the small intestine, which, in the stomach and small intestine. So that's immediately after the food is swallowed, enters the stomach and small intestine after that. This is a schematic view of what the small intestine looks like anatomically. So it's a tube, as we showed on the previous cartoon, and there are all these cells that assist in absorbing nutrients. So the fat and the protein and the sugar cross this wall that we were talking about earlier. They cross what's called the mucosal barrier. And they enter the bloodstream. So this is a cross section of the wall. These are blood vessels that drain the intestinal tissue. So the glucose you consume, for example, gets pumped right into the bloodstream. And then you can use it for your own cells and cellular functions. So this process occurs in the small intestine, the upper portion of the gut. Oh, sure, yeah.
Are those little cells, that they're wrapping around before they go across, are those the cilia that are missing, the person with cilia?
Yes, yeah. So these protrusions increase the surface area of this wall, because the more jutting out, sort of folds that are in the tissue, the longer the actual wall is, the more cells that are available for absorbing nutrients. Those projections, and then it's not shown here, but even every single mini projection has its own projections to further increase the surface area. Those projections are destroyed when there's inflammation in the gut, and celiac disease is one example of where gut architecture is destroyed. Yeah, so that process involving those cilia, if the individual is healthy and doesn't have gut inflammation. This process effectively absorbs those components of our diet. But dietary fiber is sort of the last remaining component, and it does not get broken down or absorbed. So this is key because dietary fiber is one of the main reasons that we have 3 pounds of bacteria in our intestine. Plant polysaccharides or carbohydrates that are not starch or simple sugars, that material cannot be broken down by our own enzymes. In the stomach, in the small intestine, it survives. It's very tough. And so it ends up being the main sort of chemical constituent of the material that makes it to our colon. So this is an image of a human colon. This is a barium radioscopic image. So this radio-opaque barium molecule has been injected or administered. And then it fills the different pockets in our colon, and then we can image and see the all of the locations in an X-ray image. So you can see this outline of the colon here. And this is where dietary fiber ends up, and bacteria in the intestine have evolved to break down these complex plant with polysaccharides. So the dietary fiber you eat is feeding the bacteria that thrive in your lower GI tract, the colon and the rectum. So this material, if we add it to this diagram, passes all the way through the small intestine and ends up in the large intestine. Okay, any questions on that? So I would argue that that's one of the main reasons why we have so many species of bacteria in our guts, and it's an important lever to actually manipulate which bacteria are present in our gut. Because if nutrients are one of the main benefits, by changing the types of dietary fiber that we eat, we can benefit one species versus another species in the intestine. So that's part of what my lab's particularly interested in is how do different types of dietary fiber benefit different species in the gut. Okay, so this is this idea that a lot of the bacteria are breaking down plant carbohydrates can be illustrated when you look across evolution at how different types of intestines developed. So I just pulled out some examples here. These are diagrams of the GI tract showing the stomach, the small intestine, which is usually long, and then the colon and rectum. So these diagrams are shown for different species of mammals. And these 3 species, although they are not particularly closely related, this is a monkey, this is a sheep, so those are very different animals. They share this feature of a really large stomach. So there's a large stomach here, a very large stomach in the sheep, and then in llamas also a pretty sizable chamber in the stomach. And these chambers contain a lot of bacteria that break down the plant materials that these animals eat. So you've probably heard of ruminants or cows that have a special stomach. It's the first stomach in a cow that contains a lot of these bacteria that allow the cow to break down really tough and difficult to digest foods like grass.
Michael, and
Yeah.
I mean, that's surprising to me because I always thought that the stomach was so acidic that they didn't really survive there. So these are very acidophile bacteria in there?
Good question. Yeah, I think that's a question I do not know the answer to. My guess is that the acidity of these specialized stomachs in these herbivores is actually not as acidic as an animal like a human. So we have a pretty acidic stomach, pH 2, that will kill many bacteria. I am almost positive that the acidity of these types of herbivores is less, allowing microbes to survive. It's an interesting idea. Maybe there are some acid tolerance mechanisms in bacteria that live here. That's a possibility too. Okay, so these animals are called foregut fermenters for that reason, that they have a super large stomach that immediately allows them to digest the foods they're eating. So you can contrast that with these three animals. Again, 3 herbivores, again, very distantly related. Orangutans, a great ape, pony, horses look similar, and rhinoceros. The stomachs are smaller in these animals. What's large is the colon. So as you could probably guess, this is where these animals house all their bacteria. So these are called hindgut fermenters. And they're, especially in the case of animals like horses, the chamber is enormous. They have a huge, large intestine where all the bacteria reside and break down the material that the animal is consuming, mostly the plant polysaccharides. So I think this is incredible because it shows how even very distantly related animals have this sort of convergent evolution. They end up looking very similar based on where their gut microbes are located. So gut microbes are controlling the anatomy or the physiology of these animals over evolutionary time, which I think is fascinating.
Talking about acidity, what's the role of bile? Is that just to change the pH downstream so that it doesn't cause damage?
Yeah, so for those who don't think about this every day, bile acids come from the gallbladder, and they have several functions, and a lot of their functions impact gut microbes in exciting ways. So there are many labs that study gut microbes who focus on bile, actually. Bile can be toxic to bacteria, can be bactericidal. Bile can also serve as a nutrient for bacteria, and it kind of depends on the species. A species of bacteria that has genes to detoxify bile and then convert it into nutrients, that species is going to benefit from bile, whereas a species that doesn't have those genes may be killed by the detergent action of bile in that case. So it kind of depends on the species, I would say. Yeah. Okay, so then what about us? So if we think about not just herbivores, but omnivores, here's an example of the sort of typical GI tract layout in omnivores. So rats and mice are a very common model organism we study, and they have most of their bacteria concentrated in the large intestine in a compartment called the cecum. It's sort of a blind-ended compartment, like a cul-de-sac, and that contains most of their gut bacteria, and it allows them to break down plant carbohydrates. Pigs and humans look actually a lot more similar to each other and have a relatively small stomach with a simple small intestine. And then in the colon of pigs and humans, there are all these like mini chambers called haustra that can serve as pockets for bacteria to colonize, reside there, and break down the dietary fiber that we eat. Okay, so that's sort of the situation for omnivores like us, for humans. Okay, so there was a question earlier about how do we know all of the different species in a microbial community? So in our intestine or in our mouth, how can we determine which species are there? And so the way that this has been done more recently is through what's called high throughput sequencing. So I'm not sure how much sort of molecular biology you guys went over in the previous lecture.
They're all pros now.
So I can give you sort of the details, but also just the general idea, the basic idea of how this works. We use so every bacterium has a ribosome to make proteins, and in that ribosome there are RNA molecules that are very conserved. This is an ancient protein complex. The first living organism had ribosomes, and so we haven't really changed ribosomes that much over evolutionary time. So every bacterium has ribosomes. Every bacterium has a piece of the ribosome called the 16S rRNA gene. And because it's so similar from bacteria to bacteria, we can use it as a handle to grab on to that molecule, no matter which species of bacteria is present. So we do that with what's called a primer, which is a small piece of DNA that binds to a conserved portion of the gene. And so in this image, this is just the full sequence of that piece of RNA that is part of the ribosome. And the sequence is not important, but the colors are. These gray colors are very conserved. So every species, from E. coli that causes dangerous infections, to Salmonella, to friendly bacteria that colonize a cow's rumen, to probiotics like lactobacillus, components of yogurt, fermented foods, they all have this AAUGCGG. It's the same in every species. And because of that, you can make a DNA molecule, a primer, that sticks to that portion in every single species of bacteria. You use that as a handle to make copies of a portion of the gene that is different from species to species. So there's a small portion of this RNA molecule that is called the barcode of life, because every single species has a different sequence in that region.
Can you say that one more time?
Yeah, so we can take advantage of the part of this sequence that's the same in every organism to grab onto it and then determine the part that's different in every organism, which is shown in pink. That's the variable region. And so when we make that copy, we can catalog what the sequence is through high throughput sequencing, which I won't go into. But essentially, you can learn which sequences are present in your sample containing many bacteria and count them. So this is a representation of all of the different sequences that might be present in a fecal sample from a human. And let's say you see this red sequence present. You see a green version, which just has different nucleotides. Maybe the green version is a streptococcus or staphylococcus. The red one might be E. coli. The blue one might be Salmonella. So you can see those sequences. And so you know this person has staph, E. coli, and Salmonella in their fecal sample. What's more, if there's more staph or if there's more Salmonella in that person's species, you will get more sequences. So if the blue is Salmonella, you see that this person has a lot of Salmonella in their feces. And it's quantitative. So in this way, you can actually count how many of each species is in the sample that you're analyzing. I hope that makes sense. So here's a representation of all the bacteria that might exist in a fecal sample. And then you learn by sequencing how many of each type of species is there.
I'm confused what you're sequencing for.
Yeah, so when you do the sequencing, you rely as a handle, you rely on this conserved region, which is the same in every bacterial species. So you sequence for this small stretch of nucleotides, which is known as the barcode of life. So that's how you actually, that's actually what you direct your question at. That's the piece of the DNA that you're trying to sequence, is that barcode of life.
And this is part of the 16S, that portion of the ribosome that's conserved.
That's right. Yeah. Yeah, so every bacterium has a ribosome. Every bacterium has a 16S gene. And every bacterium, a little sequence of that 16S gene is the same across every bacterium. And then the piece immediately following that part that's the same is different depending on which species it is. And that's how you learn which species is there. Okay, so so the bottom line is that we are capable now of taking anybody's fecal sample. I keep saying fecal sample because that's the easiest sample to get for studying the gut microbiota. So it's non-invasive. So there are many, millions of people now on Earth have had their fecal samples sequenced to learn which bacteria are there. Right. So we have done this, and so we have a pretty good picture of who's there, of which species are present in healthy adult humans. So I just wanted to show you some of this data and what it looks like. So first I'll go through how to read this plot. This is a plot where each bar that I've kind of highlighted with black arrows here represents a single adult human. And they provided a sample from their given body site to be sequenced to learn which bacteria are there. And the bacteria are color-coded in a stacked bar plot. And so in this person, they have a lot of this bacterium that's color-coded in yellow, and a lot of the bacteria that's color-coded in greenish gray, but very little of this red color-coded bacterium. And then if you look at this person, they have actually less of the green bacteria and a lot more of this red bacterium. And this person, again, has more red, some yellow, but even less green. And this person has almost, we're getting to very low levels of the green bacterium. This man or woman here has almost 100% of the bacterium in their sample. Almost 100% is this green bacterium. So what these type of plots illustrate is how variable it is from person to person, even among healthy adult humans in the US. You have some people with a very different collection of species than someone else. Does that kind of make sense? Or are there questions about that? Yeah.
Is that related to dietary intake?
Yeah, great question. The data that was collected on these people did not include, for example, what they were eating at the time of sampling. But there have been other experiments since then to help answer that question. And I think I actually have one on the next slide, and I'll show you. It matters what you eat. That changes which species are there. Yeah.
So does this chart suggest that some individuals have much more bacteria in their fecal sample than others? Is it quantitative?
Yeah, so that the total amount of bacteria is not plotted in this type of plot because it's just a percentage. So this person just has more percentage of this yellow bacterium, which is like E. coli, than this person all the way over here, which has mostly green bacterium.
But why is that? Why does it rise from left to right? It feels like you could represent the percentages having it be equal with the colors.
Yeah, so I think if they incorporated the total amount of bacteria in the sample, it would provide more information. But the percentage plot is agnostic to the total amount. It doesn't describe the total amount. this rise only has to do with how they've sorted these bars in the graph. So they decided to sort these people based on who had the most green bacteria all the way to the person who had the least, which is why you see the rise. Yeah, so, and as was pointed out, this is actually the mouth, the buccal mucosa. The picture of which species are there was very different depending on the body site. So the oral microbiota here, including buccal mucosa, plaque, tongue dorsum, these body sites have a lot of what you might call diversity. There's a lot of yellow, blue, green, and red and purple in all of these sites. That's a different situation than if you look over here at, for example, the vaginal microbiota or the stool microbiota or the skin microbiota. Here you can see a lot of red, and many people have, over 80% of their skin microbiota is dominated by this one type of bacteria. In the feces, it's even more sort of dominated by green or blue in these plots. So you can see that the body site matters in terms of which bacteria are going to colonize there. That's probably due to some of the benefits that we talked about before. Different species are adapted to different sites. Okay, and then I did want to just give some brief points to remember about the major types of bacteria that are present at these different sites. So you can see from these bar plots that there's a lot of green, red, blue, and yellow. Those turned out to be some of the most abundant phyla that are present on the human body. It's just a small slice of the diversity of bacteria that exists in the environment, in the world at large. These end up being very specific types of organisms that colonize our bodies. They're really adapted to us and have been for many, many years. So who are they? There are 4 major classes. So one is called the Bacteroides. I'll just say a few things about these. They are generalists, so they can consume a lot of different types of dietary fiber that we eat. And they make what are called short-chain fatty acids, which is part of the reason that the intestine smells bad. These short-chain fatty acids are volatile and they smell bad. We probably evolved to perceive them as a negative odor in order to avoid getting infected by bacteria. But they actually provide benefits to our intestine when they're produced. They're made as a result of the bacteria breaking down plant fiber. So if you take, for example, certain fiber supplements, many people have, more fermentative activity, more bacterial activity, and it can cause, symptoms that are unpleasant. Some of those symptoms involve, short-chain fatty acids and fermentation by the bacteria. These microbes are aerotolerant, which means they can survive in oxygen, but they don't actually grow in oxygen. So this is an important feature of the intestine, is that there's very little oxygen in our intestine. And so the bacteria that survive there and do really well there are often bacteria that they're anaerobic. They thrive in oxygen-poor environments. So that raises the question of how bacteria ever get out. So how does a species spread from one person to another, whether it's beneficial or harmful? These Bacteroides are aerotolerant, so they can survive short periods. That probably helps them to spread from one person to another. Okay, firmicutes is the large green slice of the pie, the large green bars I was showing on the previous slide. These include organisms that can form spores. A spore is a very environmentally resistant form of bacteria. And so spores are what help firmicutes spread from one person to another. And firmicutes are actually, classes of firmicutes are the ones that help ruminants break down the very difficult to digest material that ruminants can survive on, like grasses and hay. Proteobacteria are contain a lot of the bad actors that you're familiar with. So these include E. coli, Salmonella. A lot of gut infections are caused by Proteobacteria species. Cholera is another one. And they are, importantly, they are adapted to thrive in oxygen low environment, but can also thrive in oxygen high environment. So that probably provides an incentive for them to invade the tissues where there's a lot of oxygen. Your bloodstream is full of oxygen. So this is probably why these types of bacteria can sometimes evolve to become pathogens and invade the tissue. And then the last group that's pretty abundant is actinobacteria. These species are not really found in the gut of adults, but they're really dominant in the guts of infants. So healthy infant humans have a lot of what's called bifidobacteria. And these bacteria are adapted to consume milk carbohydrates from the milk of the mother. And so when babies are breastfeeding, their intestine is really dominated by bifidobacteria. And a lot of probiotics for infants that are being developed right now are bifidobacteria species. Yeah.
G plus, is that like gram positive?
Yeah, yeah. So yeah, the gram positive or gram negative refers to the cell wall of the bacteria. And these species have varying cell wall compositions.
And that relates to whether or not they absorb a certain stain.
True, yeah. Yeah, so the gram stain is a well-established technique for distinguishing different species of bacteria from each other. These are incredibly distantly related. So gram-positive bacteria here, gram-negative bacteria here are way more distantly related than, for example, humans and a sea sponge, for example. Animals know nothing about diversification compared to bacteria. These are very ancient splits in the biology.
So some of these are archaea?
There are very few archaea in the human gut. There's one that's present in at some abundance in some fraction of people called Methanobrevibacter, but it's really a minor constituent. And it's not clear yet whether your biology, your health will be different if you have that archaea versus if you don't. But folks are looking at that. Yeah.
If you've gotten to it yet, but your gut makes a lot of serotonin.
Yeah.
Resources.
Yeah. There's some very recent work on this and whether bacteria can stimulate the production of serotonin in the gut, whether the particular species that you harbor controls how much serotonin is made, or whether the bacteria can actually consume the serotonin that's produced. Both of those ideas are being looked at right now.
So can certain foods increase your serotonin?
That's a good question. I don't know the answer to that.
Antidepressants out of business.
Oh, yeah. Yeah, I think I do know some examples. It's not my particular area, but I know some examples where folks are trying to understand whether bacteria can make hormones or other molecules that can circulate in the body and influence other tissues. I think it's an exciting area. Yeah, and it brings up a point that I like to make as well about how you might use bacteria as a therapeutic. If we can engineer bacteria to produce some therapeutic molecule or some molecule to control our biology, one of the main benefits is that that therapy could be permanent. Unlike a drug where you have to take it over and over again, if you can colonize a patient with a bacterium that produces a beneficial molecule of interest, that treatment could be permanent. It could last as long as that bacterium survives. You might not be able to take antibiotics because then you would destroy the therapeutic. Maybe you have to take it again after you take antibiotics. And you have to make sure it's safe because you wouldn't want to engineer a live therapeutic and have it behave in a way that's detrimental. But I think there's a lot of excitement in that idea.
I think there's also a fairly famous experiment where they transpose fecal matter between, I think it was knockout mice, between fat and thin, and it changed their weight.
Yeah,
Well, really.
yeah,
Say what that was, I could not.
yeah. So I'm sorry, what's your name?
Yeah.
Anne? So Anne was talking about a famous experiment done in the 2012, 2013 era where lean mice and thin mice were analyzed to see which bacteria were present. And then by transplanting bacteria from obese mice, did I say lean? Lean and obese
Yeah.
mice.
This is the same group.
Right. So by transplanting the gut bacteria of obese mice into lean mice, the researchers demonstrated that that resulted in weight gain in the mice. So yeah, I think trying to understand how we can introduce bacteria to improve health is a really hot area. Autism, there have been a lot of studies on whether the bacteria in your intestine affect your behavior and mood. Those experiments are ongoing. So there's some controversy about whether there's an effect there. How big is the effect? The effect on metabolism, I think, has more weight of evidence behind it at this point. But yeah. Yeah.
Does it take a long time to make these changes? Is it something that you could, or that you can change for a specific reason and then change back?
Yeah, great question. So we work on this a little bit in my lab. We feed the mice. We work with mouse models, and we will feed our mice dietary fibers that humans consume, like psyllium fiber, metamucil, or citrus pectin fiber. And what we've seen is that the microbiota, the bacteria in the intestine, changes within 3 to 4 days. So it's a pretty rapid alteration in the gut microbiota. Then when you remove the fiber, you put the mice back on their normal diet, the changes revert to the situation that was present before they were eating the fiber. So yeah, I think, yeah, we're excited about how you can fine tune what's in the bacteria in the gut by what you feed them.
And there are lots of places where you can try. It seems like.
You mean places like, what do you mean by places?
Each place within where the mouse is put together and what it
True.
contains.
Yeah.
It's like a car parts. It's like, oh, that wasn't the air pump. It must be the, something else, a new generator.
Yeah.
Something.
Yeah.
You can see the end things, but there are different pathways to the same result.
Yeah, and I think the different sites within the intestine, like controlling where bacteria are in the intestine, whether they're in the first part of the large intestine or the second part, how do they interact with other species in those two regions is also something we're pretty interested in too. Yeah. Okay, so I know we're running a little low on time, right? It's 12. Yeah, so I wanted to just leave you with one additional idea, which has to do with how we know all these things about the gut microbiota, how we know they could affect weight gain or immune health. And so I'll skip to that for the last slide here. If anyone's squeamish, there's some pictures of mouse dissections on the next slide, so you can look down. But the way that we study this in my lab, but also in many labs, is through the use of what are called germ-free mice. All of us are born with no microbes. We're immediately colonized when we're born. And so and even if we take antibiotics, there are still bacteria in our intestine that survive the antibiotics, even though most of them die. So it becomes really difficult to study this in humans. And what we can do using an animal model is to create a situation where there's no microbes. It's as if there were no bacteria on Earth. These mice are born into these sterile bubbles, and all the food and bedding and water that we provide them has to be sterilized, so there's no bacteria inside. If we even allow one cell, live bacterium into this environment, it will colonize the mice and immediately take over the gut. There's no competition from other species, so it just fills the gut of the animals. They're fine, but then we can't interpret the experiment. So we raise the mice in a totally sterile environment, and then we can compare what is life like if you have no microbes in your gut versus if you do have normal microbial species there. And we've learned these things, and I'll just kind of share these with you, and then we can stop for questions maybe right before that. So the main things that we see, first of all, we see that the mice that have no microbes, germ-free mice, have an enormous cecum. Their large intestine is huge compared to mice that have bacteria. It's a little bit counterintuitive. But the reason is because the bacteria there's no bacteria to break down the material that's in the diet of these mice. So the material builds up in the cecum. And there are no germ-free humans, but if there were, you could hypothesize that we might have large changes to our gut physiology if we didn't have microbes to break down the foods that we're eating. So that's one thing. The second thing has to do with body fat. So mice that have no microbes have really, really low levels of body fat. In white bars are the germ-free mice. In black bars are mice that have a normal microbiota. And then this is a 4-week animal and an 8-month-old animal. You can see that over time, mice get fat, they develop fat. But if they are germ-free, the fat is way down. This is despite the fact that they consume more food as well. They consume 1.5 times as much food as mice with bacteria. And a lot of people are working on this right now for obvious reasons. It's very interesting how bacteria are controlling fat deposition and weight gain. Germ-free mice need to have vitamins added to their food, extra vitamins, because bacteria in our gut synthesize vitamins that we need. And so if there are no bacteria, you need to provide extra vitamins. Otherwise, the mice they won't survive, they'll die. They have a skewed immune system. So a lot of people are interested in this as well. Germ-free mice have less regulatory T cells, which are anti-inflammatory T cells in their gut. And they have other immune alterations that are very interesting. And then the last piece is their susceptibility to infections by pathogenic microbes. And this is one of the main, I think, evolutionary benefits for humans and mice and mammals in terms of why they're colonized with gut bacteria. So if there are bacteria present, they take up space, they outcompete pathogens that would come in. If there are no bacteria present, pathogens have no competitors, and the mice are incredibly susceptible to gut infections. And we see that in people actually too. So if you take antibiotics, you have increased susceptibility to many infections, including Clostridium difficile infection. And so I'll leave you, since somebody had mentioned fecal transplants. This is a powerful demonstration of the importance of gut microbes. If you take antibiotics and you become infected with Clostridium difficile spores in the hospital, the current best treatment for that infection is to receive a fecal transplant from a healthy human, because these microbes in the fecal transplant will suppress the Clostridium difficile in a way that antibiotics just can't. So this obviously has a lot of downsides as well, and there's a lot of concern about where you get the fecal material. It needs to be a healthy donor. And so companies are trying to standardize this, maybe just grow the species that you need instead of harvesting them from a donor. But currently there are no products yet. There's no Clostridium difficile cure that has replaced fecal transplants.
Going back to the mice, you said that it, the no bacteria mice were very susceptible to disease. And then you said it was only intestinal disease. What about other disease, respiratory and
Oh, yeah.
other diseases?
That's a good question. I don't know the literature enough to know. So other diseases like respiratory disease might be viral, like flu or pneumonia, bacterial pneumonia.
Or TB?
Yeah, TB. Off the top of my head, I'm not recalling any really dramatic effects in the papers that I've read. I think the idea that the gut microbes actually outcompete a pathogen that's in the gut is an important way that operates. It's called colonization resistance. Yeah, so I don't know of other examples. Actually, there's an example from skin where skin bacteria outcompete commensal skin bacteria outcompete pathogenic skin bacteria.
That's kind of the same.
Yeah, that's a similar idea.
Versus like.
Yeah.
Because I've seen in kind of health food store type stuff about how you're gonna boost your immune system by taking these whatevers. And the whole idea is all diseases is what the advertising seems to imply.
Yeah. Yeah, and I think there's a lot we don't know still about how that might work, how changes to the gut microbiota would affect a distant tissue site.
Yeah.
I think that's, if that occurs, it's probably more complicated, harder to understand.
Because so that could be like false advertising.
Potentially, yeah, you do have to be cautious, right, about what claims are made, especially with a supplement, dietary supplement, or with anything that's not FDA approved or has had a clinical trial done. The fecal transplant in terms of Clostridium difficile has had a lot of clinical trials, so we feel pretty confident in that treatment. But yeah, my hope is that there's more research and we can do more human studies so that we can really figure out which supplements and probiotics have a big impact and which ones maybe can't make those claims. Yeah.
When you're showing the bacterial percentages for the gut from the gut sequencing of normal healthy individuals, it immediately came to mind what about looking at the sequences, the sequencing of the gut microbes of unhealthy people, people with particular disease conditions? Is there someone that's doing in-depth research of what those differences are?
Yes, yeah, a lot of research on that. The results usually involve describing an imbalance to the bacteria. Often it's called dysbiosis. It's very rare to be able to pick out a single species that is causative. So to point to everyone with type 2 diabetes or everyone who's suffering from a tuberculosis, susceptibility to tuberculosis, to be able to point to one species in the microbiota of those people as causative. So that's been really difficult. Often it's impossible. There's no one species that we can point to. But what is clear is that the balance is significantly different. The balance of all the species is different than the balance that you see in healthy adults. And then the question becomes, is that correlation or is that causation? Is the disease causing the bacteria to be adjusted, or are the bacteria participating? Are they changing the disease? So that's what we need to figure out. So the germ-free mice is one way to test that, because you're adding in the bacteria of interest. That's the result that you get is going to be the cause of adding in those bacteria. Yeah.
I saw on one of your slides mention cyanobacteria in the gut, I think.
Oh, yeah, I think
I was just kind of curious. My understanding is those are among the most primitive bacteria and create oxygen. But there's no oxygen in the gut either. But is there any comment on that, or am I on the wrong
No, I mean, definitely it's here, in light green, and so the amount is very small.
Oh,
Um,
okay, I was looking at the wrong green. then.
yeah, I actually added this to try and clarify, but I made it more confusing. I think we have some projects in the lab looking at cyanobacteria, because we think they could be an interesting way to modulate gut microbes. They're often very rare in the gut, which is why they might be a good way to manipulate gut microbes in many people. You won't have a interference of cyanobacteria that are already there. Some people, I think, believe that this may be due to the diet. So if the person is eating, for example, spirulina or algae supplements in their diet, that would maybe show up in their stool, for example. I guess it wouldn't show up in their airways or on their skin. But yeah, pretty low. But I think there's some interesting things to look at.
Right. So I assume underlying Roger's questions, these are photosynthetic normally, right?
Right.
So they
Yeah,
shouldn't-
so
yeah,
there's
don't get a lot of sunlight.
Right. But yeah, we've been curious about transparent organisms. So sea slugs, for example, can be colonized by photosynthetic bacteria, or there are fish that are clear. And so I think it'd be really interesting if we could discover commensal bacteria that colonize the gut of animals where light can reach the gut. Yeah. Okay, well, I think hopefully that was interesting, and I'm happy to answer additional questions too by email if that'd be helpful.
Right. Well, thank you very much. All right, but I'll bet there are lots of questions. So let's take a few minutes. I certainly have questions. Other people have questions.
Oh yeah, if there are more questions also, yeah, I'm happy to.
Well, my
Yes, sir.
question is, when you take an antibiotic and you wipe out most of your intestinal flora, it's surprising to me that you usually don't feel sick. It suggests you can really function pretty well without them. I mean, what's known about that?
Yeah. I think that's pretty important to consider. I guess the short term administration of antibiotics is probably going to have less of a noticeable effect than the long term.
Right.
The understanding how many of the bacteria you actually deplete when you take one antibiotic versus the other is another important consideration. Oh yeah.
I've had frequent bouts of diverticulitis, and one of them was caused by taking amoxicillin from tooth extraction.
Oh yeah.
That did not take out the bacteria from the diverticulitis, but took out everything else that was keeping it suppressed.
Yeah.
I ended up in the hospital with that one, tooth extraction.
Yeah, that's a similar scenario for Clostridium difficile infection. Yeah.
I read a little article it was a newsletter from Caltech a couple of years ago about association between
Bladder mark.
between Parkinson's and gut biota. Do you know anything or more research on that?
Yeah, one of my colleagues at UC Irvine is very interested in how the presence of certain species in the gut could potentially cause aggregation of proteins at distant sites. I think that's very recent research too. So it's exciting, but we probably can't say for sure whether it's going to affect Parkinson's. Parkinson's is such a strong genetic component that, yeah, I wonder how much microbiota bacteria is an environmental component, right? You inherit it from the environment. So I wonder what the contributions would be.
Right. Let me just ask, are any of the Zoomers have questions? Now's a good chance here. Okay.
No.
I guess not. Michael, do any of these major classes make methane? Where does our methane?
Oh, interesting question. Yeah. There are definitely methanogens in the gut, right? So the gas that comes out of the intestine contains a lot of methane as well. That's part of the odor, in addition to sulfur containing compounds that the bacteria make. I want to say the archaea, so the Methanobrevibacter, is a methanogen. But I think there are also bacteria as well that produce methane.
All right, well, okay, couple more and then we'll.
Oh, yeah.
close.
Are there any sort of just graphs or straightforward things that show which food you eat encourage the growth of which bacteria? I know
Yeah.
it wouldn't be a one-to-one, but category wise.
Yeah, I wish there, yeah. We've tried to do experiments in a controlled setting with the mice that we're feeding fiber to answer that question, to be able to map which food, which types of fiber will boost this species or this species. But it's been really difficult. Yeah, I think it is, the difficulty comes from the differences between each person and how I didn't mention it, but these two people that both have green firmicutes bacteria at a similar level probably have different strains of firmicutes with different properties. So the diversity from person to person is even more exaggerated than what it appears here. And so it's been really hard for labs or companies or health professionals to accurately say this type of diet will bring you to this microbiota configuration that's helpful. So that's what we need to try and understand, but it's been hard.
Okay, one last one, probiotics. What do you think? I mean, a lot of them just only contain 1 or 2 bacteria. Is there anything out there that is actually worthwhile?
Yeah, I would say that I'm really excited about probiotics where we know the mechanism of how they're going to act in the gut. I think if probiotics are working for you, then you should, continue to use probiotics, or if your physicians are, advising you to try this or that probiotic, you should follow their advice. I think we need to know more. A lot of probiotics are not FDA approved drugs, right? And there's a reason for that. We just don't know with as much certainty whether they are going to help a broad swath of people.
All right, well, thank you very much.
Well, related to that, so it would be more advantageous to take a probiotic that says like 50 billion versus 100.
Yeah, there's a lot of regulation, I think, in terms of the claims that, companies can make about their probiotics. Considerations for like the number is whether that number represents live organisms, or is it just, is it bacteria that are mostly dead? Are they,
They're controlled regarding refrigerated versus non-refrigerated.
Yeah. I think it's unfortunately pretty difficult at this point to make those judgments as a consumer about which probiotics are going to be most helpful. So we're kind of left with a trial and error approach.
Okay, next week, please return. Another speaker.
Thank you.